So I am Charlie Monica, in case you don't know me yet. As Julia mentioned last week, Overdue and I will be helping to organize the Brain Map starting this year. So if you have any potential speakers that you would like to invite, please don't hesitate to contact us. So today it's a great pleasure for me to introduce you Dr. Margaret Livingstone. Margaret is a professor in neurobiology at Harvard University. She went to MIT for her undergraduate and received her PhD and postdoctoral training in Harvard. Margaret is a neurophysiologist, and she is best known for her work on visual processing. She did groundbreaking work on parallel processing of visual information. She discovered a new subdivision within the visual system for color, form, motion, and depth, and recently she applied fMRI in non-human primates to help target the single unit recording, and has applied the technique to study face perception. Another interesting area of her research, although it's probably not the focus of the talk today, is to apply what we know about vision to study visual art. So the topic of her talk today is apparently different to what she sent me last time. So the topic is behavioral expertise and domain formation as a consequence of early but not late symbol training. So welcome.
That's the dry title. That's what we're really going to talk about. So I suppose most of you do work on functional modules in mostly humans, I suppose. Are there any monkey people here? No? Okay. All right, good. Some monkey people. So this is a little loud. In both humans and monkeys, the brain is divided into these two major streams, the dorsal stream, which carries information about position and how you interact with your environment, and the ventral stream, which is for object recognition. And we're going to talk about modules in the ventral stream. So if people get lesions in their ventral stream, they get what are called agnosias, where they lose the ability to recognize specific categories of objects. I'm sure you all know this, but you can lose surprisingly specific categories. So neurologists have discovered that you can lose the ability to recognize small manipulable objects or large objects or faces, prosopagnosia, or animate objects, or writing, text, or body parts. So the fact that you can specifically lose any one of these categories means that there must be parts of your temporal lobe dedicated to processing those categories. Those are the modules I'm talking about. And people with agnosias can actually copy objects. This is a patient copying these things who couldn't recognize these things. So vision is unaffected, but your ability to recognize things is totally abolished. We know in monkeys from electrophysiology that there's a hierarchy going from the posterior to the anterior of the temporal lobe, with simple kinds of receptive fields more posterior and more complicated kinds of receptive fields more anterior. And Grill-Spector did a lovely study showing that as you go more anterior from V1 to LO, the responses to intact objects are more anterior and to parts of objects more posterior. So there's a hierarchy of object complexity along the temporal lobe until you get to things like the Jennifer Aniston cells in way anterior temporal cortex, so cells that will recognize one individual in any manifestation. So I want to talk in particular about faces, language, and reading, because these are all processes that are localized, and I want to see if we can talk about the mechanisms, what having these kinds of modules does for you. So we know that language is localized in humans, faces are localized in both humans and monkeys, and reading is localized in humans, and oddly, reading is localized to just about the same part of the temporal lobe as faces, but in the opposite hemisphere. And we know that there are behavioral or sensitive, there are critical periods or sensitive periods for all kinds of things for which we have modules, like the ability to recognize faces, language, reading, imprinting, learning Braille, texting, and song. All these things show windows in development when you can learn them better than at other times. So, for example, we know that language shows a sensitive period because children learn languages perfectly, but adults, you can spend decades immersed in another language and never get rid of your accent, right? This is also true of face processing. So there is this phenomenon called the other race effect. You probably have more trouble learning faces of an ethnic group that you were not brought up with. Many people consider this racist and are kind of ashamed of it, but it turns out it's not racism, it's just who you grew up with. You, just like language, learning to recognize other faces has an early developmental window. So there are studies to show this, but I find it more compelling to, I met this kid named Adam, who was a friend of my son's. And Adam is a white kid who grew up in Jamaica. And when I met him, I said, "Oh, when you first came to the U.S., did you have trouble recognizing white people?" And he acted like I had just figured out some deep problem, social problem that he had. So I have to remember to turn this on. Now, I know what I'm doing. Watch this. I know what I'm doing. This is Adam.
I grew up in Irish town, Jamaica, which is about an hour and a half outside of Houston. And when I first came to America, especially with girls, I could not tell the difference between white girls. It worked out to be quite problematic when I started dating one of them. But they all seemed quite the same.
He was very happy to find out that this was something he maybe could get over. So when you learn languages, sorry about that. When you learn languages, you can use preferential looking with infants and find out that they prefer to listen to vocalizations more than noise. But it doesn't matter whether the vocalization is a monkey or a human. They'll listen to either one equally. But then at about 3 months old, they start preferring to listen to human vocalization compared to a monkey, and then language studies have shown that they start learning to like the phonemes of the language that they're hearing, and then after a while, of course, they learn to speak their own language. Same thing's true in faces. So infants, very young infants they can tell the difference between different monkey faces, because if you show them one monkey face and then another, they can tell that it's novel. But when they get to be about 6 to 9 months old, they can still do it with people faces, but they can't do it with monkey faces. They lose the ability. So although there are clearly developmental stages in learning faces and learning language, there's a strong idea that these things must be innate. So, for example, infants, both monkeys and people, will look at a face preferentially. So this is further the idea that there's something innate about face processing. So despite the fact that they show a sensitive period, there's a strong idea that face processing must somehow be innate. That is, there's a strong genetic component to it. And of course, there are some very intelligent people who have contended that there's something innate about language. Although you learn your own language, there's some special module that must be involved in learning languages. But there is no way that reading is innate. Humans have not been reading universally long enough to have evolved a reading module. Yet we have a reading module. So we wanted to ask about monkeys, because you can manipulate their environment legally and ask how they I'm supposed to turn on the mute here. So monkeys recognize faces like us, primates, They use faces for social interactions and for recognizing conspecifics. And as I mentioned, like us, monkeys have special regions in their temporal lobe, just like we do, that are selectively activated when you look at faces. But we didn't want to ask about faces in monkeys. We wanted to ask about symbols. We wanted to find out whether, like people, monkeys could develop an area selective for artificial shapes that there's no way they could have evolved a selectivity for. So we got 10 rhesus macaque monkeys. 4 of them were prepubertal, that is, they were juveniles, and 6 of them were adults. And we taught them these symbols. The NIH wanted to know what theoretical basis I had for teaching them these symbols. I had none. I just thought, these are the symbols that we use. Culture has somehow adapted them so that we can easily recognize them. They're easily discriminated. So I thought, why don't we just take advantage of this instead of having some theoretical basis for these particular shapes? Why don't we use ones that we know that primates can learn? So we did. Oh, I'm not supposed to turn off the mute now. This is Mary Todd. And Mary Todd is touching this screen in response to pairs of shapes. And so for F, she gets 17 drops, and she chose that over 1. And then the next time she gets a trial, she gets N and 8, and she chooses N over 8, and so on. So she gets however many drops correspond to each of these symbols. So they're rank ordered from 0 to 25. And despite the fact that whichever one the monkey chooses, the monkey's going to get something, except for 0, the monkeys pretty much always choose the bigger one. And they can either choose between sets of dots in a circle or 2 symbols. We taught them both automatically in their cage every day. They get a lot of experience. So we have 6 of these setups, 4 monkeys per setup, and they cycle in. They spend several hours every day in the training cage. So they get a lot of experience, 7 days a week, 3 hours a day, for years. And so we're going to display their behavior on a grid where we plot the stimulus that they chose. So it's always 2 stimuli. We plot the stimulus that they chose and the stimulus they didn't choose. So anything up in this half is going to be a larger choice, and anything down here is going to be a smaller choice. And we use color to map it. And you can see how good they are. They almost always choose the bigger one, whether it's dots or symbols, and whether it's the kids or the adults. And for those of you who are in psychology, this is the scalar variability, the Weber fraction, but the rest of you don't need to. What?
How long did it take them to learn this?
Get there. But surprising, they're really fast. The kids are faster than the adults. And it takes them, sometimes it'll take them 1 day, which is like 300 trials, to get a new one. We taught them in order. So that's actually what I'm going to talk about next. How long did it take them? Age in years, number of trials to get, so we teach them in order. We started them with 0 and 1, and then we added 2 and 3 and 4 and so on. And so the juveniles took less than 50 trials with a new symbol for each new symbol to learn it, and the adults took a lot more trials. But that's for the symbols. For the dots, everybody got the principle that the more dots, the better. And we didn't have to teach them new dot numbers. That's been shown before. So the kids were faster than the adults. And the kids were also more fluent than the adults. So if we look at the reaction times, the juveniles had about less than a half a second latency for responding to any of the dot sets or any of the symbols. The adults were just about as fast as the juveniles at responding to the dots, but they were quite a bit slower responding to the symbols. So they were less fluent than the juveniles. And I plotted it again this way. So this is symbols reaction time against the monkey's age in years, and you can see that this is the youngest adult. that there's almost a linear relationship with age and reaction time, but there's no relationship for the dots. So that means the adults can move as fast, they're as motivated as the juveniles, they understand the task as well as the juveniles, they just aren't as fluent as the juveniles. So this is the summary of our behavior. So the juvenile monkeys learned symbols faster than the adults. They responded faster to the symbols than the adults. But the adults were just as fast as the juveniles at responding to a non-symbolic task. So then we said, well, what parts of their brain are involved in this task? So we did functional MRI here. And we showed them blocks of learned, unfamiliar. So here, these are the symbols that they learned. We had a set of unfamiliar symbols. We chose Greek letters and whatever letters of the alphabet were left. The reason we used numbers, digits, and letters was because we didn't realize how fast the monkeys were going to learn this. We started with digits, and in a few weeks they got up to 9, and we thought, well, we don't want to go to double digits. That would be complicated, so let's start picking letters. If we had known that they were going to be this good, maybe we would have started with letters to begin with, but we didn't. So we have a combination of letters and digits, and then we used Greek letters as visually similar but unfamiliar symbols, and then we had faces, just as an anatomical marker. And we scanned 6 monkeys, 2 adults who had learned the symbols, 3 juveniles who had learned the symbols. Number 4 juvenile did not want to sit still in the scanner, and 1 adult who had no symbol training. The juveniles, a young monkey has a very thin skull, so many people who do monkey scanning you have to implant a head post on the top of their head to hold them still because they don't follow instructions really well, and they're okay with having a head post, but you can't do that with a juvenile monkey because their skull's too thin. So we developed a little helmet thing that we strap the monkey's head in, and there's one of our juvenile monkeys sitting there, not too upset. If you were upset, you would know it. So the 2 adults who had learned symbols late in life showed alternating regions of shape and face selectivity. So shape selectivity means that the region responded better to either of the symbol sets than to faces. And we didn't see any regions in the adults that responded more to the learned symbols than to the unfamiliar symbols, untrained symbols. So we have 3 adults who we've scanned, 2 trained and 1 untrained, and essentially the pattern is about the same. You see patches of face selectivity, usually about 3 of them, and then patches of shape selectivity. And then when we look at the juveniles, you see the same pattern of face and shape selectivity, but right about in the middle shape patch in all 3 juveniles, bilaterally in this one and unilaterally in these 2, we saw regions that were more active to the learned shapes than to the unfamiliar shapes. And here are the time courses in the adults, average time course in the adults and the juveniles. In the face regions, you can see that they have 3 different regions that respond better to faces than to either set of shapes. And then in the shape patches, we found much better responses to the shapes than to faces, but only in the juveniles did we find in the middle shape patch an area that responded more to the learned shape than to the unfamiliar shapes. And this is an overlay of all 6 monkeys of the shape regions and the face regions. And it seems like there are 3 face regions, and then just ventral to each of the, and slightly posterior to each of the face regions, there's a shape region. So there's actually, if you look at enough monkeys, there's some, yes?
I just have to know, can you just give us some details on how you define the regions? So it has to be significantly active to either stimulus, and then there has to be a...
It's a conjunction analysis. It has to be significantly more active to... This has to be significantly more active to faces than to learned shapes, and to faces than to unfamiliar shapes. And we mapped the retinotopic areas, so it turns out that each of these pairs of patches seems to lie along one of the major borders of the temporal lobe. So the V4 TEO border, the TEO TE border, and here the TE, TG border. But that one's kind of messy. There's a lot of variability there. But there's some pattern to this, although between monkey to monkey, it wasn't precise. So here are conclusions from the behavior and the functional imaging, that behaviorally, juvenile monkeys learned faster than the adults, and they responded faster to symbols, even though the adults were just as good at dots. And then all the juveniles and none of the adults showed regions that were more active to the learned symbols compared to the visually similar but untrained shapes. So now that's all the facts I'm going to tell you. These results are similar to other results that have come out about the organization of monkey temporal lobes. So again, also scanned here, Roger and Leslie Ungerleider found that there were multiple patches of face, shape, and body scattered along the temporal lobe. And they contended that these may represent animate versus inanimate categories. So, of course, faces and bodies would be animate categories, and places and shapes would be inanimate. And that's perfectly consistent with what we find, because our shapes are some kind of inanimate object category. So now, that's all the facts I'm going to tell you. And I know that it's kind of weird to spend the entire second half of a talk talking about the implications of this, but we have an idea about how our results suggest that functional modules may get there and what good they may do. So first I want to talk about what good are functional modules. So the usual idea is that you have a functional module because evolution decided that we evolved an area because whatever this category is, it's biologically important. So people think we have face modules because faces are biologically important for primates. But maybe instead of talking about whether something is innate or not, let's talk about how these things are processed. What is their processing style? And I want to talk about face processing in monkeys. This is work that Doris Sau and Vinrich Friewald did in my lab and here at the Martinos Center many years ago now. Horrifying. And what they found they used fMRI to target electrode recording to the face-selected regions in the temporal lobe. And what they found out was that physiologically, so each row is a single cell, each column is a single image type, and these are faces, body parts, fruits and vegetables, gadgets, and hands. And they found that if you record in one of the face areas, fMRI identified face areas, you find that individual cells respond much better to faces than to any other object category. That is, yes, these face patches are chock full of face-selective cells. And these face-selective cells respond to faces, they respond to human faces, they respond to monkey faces, and they even respond to simple cartoon faces. Anything you would call a face cell will respond to more or less. So that allowed us to use cartoon faces to parametrically probe how face cells code faces. So we have these cartoon faces that have 20 different parameters that involve face shape, mouth shape, nose shape, the spatial arrangement of the different parts of the face, the hair, and which way the face is facing. And we varied these all across 11 variables each. Now what we thought we were doing, there are seats up front if you want to come in. You don't have to sit out there. So this experiment was wildly successful, so I can admit now that we thought we were doing something completely different than we ended up doing. we thought we would define what makes a face. So we were trying to vary all these parameters, like outside of face space, so that we'd figure out what the critical variables were for making something a face. But we never got outside of face space, which was really a surprise to us. So this is the stimulus we used, and I just want you to see, first of all, it goes by really fast. We're varying every one of those parameters at the same time, and we're going way outside of plausible primate face space, right? Like we have cyclopean faces. The animals did not grow up seeing any of these faces, right? And yet we found that cells responded to these different parameters. So we found that each cell was tuned to about, on average, 3 of these parameters. Never tuned to all of them, but tuned to 2 or 3 of them. And you can imagine that if a cell were tuned, that is, it responds best at some particular range of that variable, you might want to have, if your cell cares about inner eye distance, you might want to have cells that peak at different inner eye distances, so you could recognize individuals based on their inner eye distance. Or you might imagine that you could have cells that responded to big inner eye distances and cells that responded to small ones, and you get everything else for free in between by the ratio of the two. And that's actually what we found, to our great surprise. So here's a typical face cell, way down here, sorry about that. This cell is tuned to 4 parameters. It cares about eye size, eye shape, and inner eye distance. But in each case, it responds to one of the extreme values of this parameter. And when we looked at, and that's typical, these are all the tunings we saw in 3 monkeys for all of the variables. And you can see that cells are either tuned to one extreme or the other, but hardly any are tuned in the middle. But mostly they're extreme tuning. We call that ramp-shaped tuning, responding to one extreme or the other of any variable. So what that says is that face cells code how a face, whatever face you're looking at, differs from the average face. And that may explain why caricatures are so powerful in evoking an identity. It's much easier to recognize a caricature of somebody than to recognize a veridical line drawing. And that's because caricatures exaggerate how a face differs from the average face, and it evokes a very strong identity. But it also means that face processing involves within category comparisons. That is, it involves local opponency within face space. And I come at this as a low-level visual physiologist, and I grew up somehow knowing that inhibition in cortex is always local. Neurons that have long axons are always excitatory. Inhibition is always local. That's just the canonical cortical circuit. So if inhibition or opponency is local in visual space, you get local inhibition in all kinds of realms because visual space is mapped onto the cortex. So whatever inhibition you have will be true to only one part of visual space. That gives you all kinds of local surround effects in luminance, motion, color, orientation, and so on. But if you have clustering of some category, it means you're going to have opponency within a category. That is, you're going to have local comparisons within that category, more so than you would have if cells coding that category were separated spatially within the cortex. So clustering in cortex gives you within category comparison, which is expertise, the ability to compare, find differences between similar objects. So we have a local opponency in all kinds of things in vision. This is what I grew up doing. You have local opponency in luminance. So as Steve Kupler discovered, if you shine a spot of light on the retina, you get a response. But if you increase the size of the spot of light, you get a worse response because you have surround opponency in luminance. That's center surround. You have surround opponency in orientation. This is vertical, but it's surrounded by a different orientation. You have surround opponency in contrast. These two blocks are identical, but they look different because they're surrounded by different contrasts. You have surround opponency in motion. That red dot was moving vertically. You have surround opponency in color. These two gray X's are the same, but this one looks less blue or more yellow. This one looks more blue or less yellow. And if you'll just look at this dot for a second, I'll show you surround opponency in color. So you're looking at some words written in white text, but you will get a yellow after image from white text because of the surround opponency. And of course you get opponent effects in face recognition. So this was discovered by Arasha Fraz and Patrick Cavanaugh. What I want you to do is look at these numbers as I cycle. You're going to be moving your eyes around, so you're not getting a local retinal adaptation. What you're going to be getting is a face identity adaptation because you're moving your eyes around. And now I'm going to show you two identical morphs. And I hope you saw a face after effect. Local opponency within a module isn't spatially opponent, but it's identity opponent. So, again, I came at this from a much different point of view than most people who work on faces. Most people who work on faces come from psychology, and they study how people process faces. I come from low-level vision, and I think about things like local opponency. So the psychologists contend that face processing is different from the processing of other kinds of objects in that it is expert. We are very good at recognizing and discriminating faces, much better than we are at discriminating objects with similar differences, physical differences. We're much better at recognizing faces. And they contend that face processing is holistic and therefore has to be carried out by a different kind of circuit than the rest of object categories. I think this is an unfortunate term, but they mean something quite precise when they say holistic. It means you have to process a face as a whole object. You can't process the parts of it. Even though you can look at the parts of faces and see eyes and noses and mouths and you think you can discriminate them, it turns out you really are not very good at this. So you can look at these two faces, and even though you know that there is this face inversion effect, and you're ready for it, and you know which of these faces is the one I've messed with, right? Even though you know this, admit it. You really saw it differently, didn't you? Yes, you can't. You can't see that the same way when you process it with non-face cortex. When it's upside down, it doesn't qualify as a face, so you don't process it with your face cortex, and it looks quite different. The other characteristic of holistic processing is that you can tell that these two halves are identical when they're offset from different bottom halves, but when you make them part of an intact face, it is very hard to tell that the top and the bottom halves are still identical. You process faces obligatorily as a whole. So one way, as I said, of interpreting these characteristics of face processing is that expert processing of some object categories comes about because evolution has endowed us with special circuitry that provides fine distinctions and is, for some reason, holistic in its nature. Or that it allows us to learn these things better than other things. But, as I said, functional clustering gives you interactions between cells that respond to the same category of objects, and that, I think, gives you expert processing because you have within category comparisons in this module. But because if you have a functional cluster, you must have a category-specific detection gate. That is, you don't get into that module unless you qualify as that category. I contend that necessarily also gives you holistic processing. Now, why do I say that? Well, if you have modules that process faces specifically, and you have parts of the brain that process non-face objects, an upside-down face isn't going to qualify as a face, so it's going to be processed by the non-face module, or a split face will be processed by the non-face module. So that would explain why upright faces are processed better or differently than inverted faces. Prosopagnosics, who have lost face cortex, don't show the face inversion effect. They're equally bad at processing right side up and upside down faces. And there's a patient, CK, who had lost object cortex, who was perfectly normal at face recognition, but he couldn't recognize inverted faces as faces. So instead of requiring some kind of special circuitry, you can explain holistic processing by having functional clustering. Of course, functional clustering is a special kind of circuitry, so the psychologists are right that face processing has a special kind of circuitry. But as a neurobiologist, I like this idea because functional clustering is a kind of circuitry that we understand already. So language is also processed holistically. Lieberman and Mattingly showed that if you play the parts of phonemes, like the formants of da and ga, if you separate them, they sound just like chirps and whistles. They don't sound like speech. And they don't sound like if you put them together, they would sound like speech. And if you add these formants back in again, the whistles suddenly start sounding like speech and stop sounding like chirps and whistles. So there's a holistic aspect to speech processing as well. And of course, even though you can look at text and see the individual strokes that make up letters, there is something holistic about reading as well. So there are dyslexics, people with strokes, acquired dyslexia, who can read whole words but can't distinguish individual letters. Stuart Anstis showed that UCSD undergraduates are inaccurate at deciding whether the top halves of simple words are identical between pairs of words when they're part of an intact word. And of course there was this email that went around a few years ago showing that first and last letters of words are key in recognition, and of course it's very hard for most people to read upside down text. So now I want to talk about how do we get functional modules. I've suggested what they're good for. How do we acquire them? So our results in juvenile monkeys indicate that early experience, and the fact that people have visual word form areas, indicates that experience can cause the formation or the refinement of clusters of neurons selected for specific object categories. In particular, in monkeys, selected for object categories that monkeys never normally use. So there was some regularity that I showed you to our face and shape domains. Roughly one pair of patches per major cortical subdivision. And of course, our results are completely consistent with the proposal that there are some innate circuits that get modified by experience. But this regular pattern of face and shape segregation suggests to me, coming from a neurobiology background, that there's some rule-based organization here. So, again, in early visual areas and early sensory areas, there's a profound effect of early experience on ocular dominance columns and visual cortex, barrel fields in somatosensory cortex, sound localization, and tonotopic maps in auditory cortex. So many, possibly all, early sensory cortices are segregated by early experience into regular, stereotyped functional domains. How could this not be true of IT? How could it be true of early visual cortex and not be true of some downstream area? So we know that neuronal activity can drive functional segregation of inputs in such a way that commonly coactive inputs cluster. So ocular dominance. Does everybody know what Hebbian mechanisms are? Come on, do you or don't you? Yes, okay. Hebbian mechanisms. Inputs that fire together tend to cluster. That makes the postsynaptic cell have particular response properties. So in primary visual cortex, you have these domains of selectivity to the right eye, left eye, right eye, left eye. Babies start out with inputs from the two eyes diffusely scattered all over the input layers, but then adults end up with right eye, left eye segregation that's quite regular. If you close one eye, you can completely lose the inputs from that eye. There's functional segregation in V2 with these cytochrome oxidase-rich stripes. That's probably activity dependent. And I think these stripes in IT may follow the same kind of stereotyped segregation that you see in V1, V2, somatosensory cortex, and so on. Now, of course, the scale of these things is much coarser than it is in V1, but Hebbian mechanisms alone, if you have Hebbian mechanisms segregating out the two eye inputs in cortex, Hebbian mechanisms alone only give you a salt and pepper segregation. So any given cell will become dominated by one eye or the other by Hebbian mechanisms, but there's nothing in a Hebbian mechanism alone that says that these two cells should have the same eye dominance. What you need in order to get ocular dominance columns is to have lateral connectivity. And once you plug in lateral connectivity, that defines the scale of the clustering of the inputs to that area. And so maybe the reason that the scale gets coarser going from V1 to V2 and then on to infratemporal cortex is that the scale of the lateral connections or the stage at which they sort out may change. I don't have any answer for this, but I think that one could come up with a rule for that. The other thing that I want to point out, and I love this because we don't know why it's true, but it clearly is true, functional domains always intersect areal borders at right angles. Ocular dominance columns intersect the V1, V2 border at right angles. In New World monkeys who do not have ocular dominance columns, orientation columns intersect the V1, V2 border at right angles. The stripes in V2 intersect the border at right angles, and these stripes intersect the borders at right angles. I don't know why, but I think it's cool. So the view we encountered when we started looking at face processing is that the biological importance of certain categories drives the evolution of specialized circuits for processing these categories in optimal ways. We propose instead that intensive early experience drives the segregation of inputs processing frequently experienced object categories, and that this segregation permits fine within category comparisons and causes their processing to be holistic. That is, we propose that instead of evolving circuits for certain categories, we evolve a mechanism for being good at responding to whatever it is we encounter. So if early experience drives functional segregation, you'd expect age of learning effects on modular organization. Like we see number areas in our monkeys. There. So, don't yell at me about this. There are some reports of age of learning effects on language. So I'm sure there's like more modern results on language localization. But apparently second learned languages are more diffuse and more anterior than first learned languages. Certainly we know from neurosurgery and from neurology that people can have strokes that affect one language and not another language. So there must be some kind of segregation of languages learned at different ages. Dehaney found that people who learned to read late in life, so he had a population of people who could read and a population of illiterates. In Portugal, in families, in fishing families, in small villages, the oldest daughter stayed home to take care of the kids. So there was a large population of women in Portugal who never learned to read, which is what they did these experiments on. Sorry, it makes me a little indignant. So in illiterates, the part of the brain that becomes the visual word form area in literate people responds to faces in illiterate people, suggesting that there is some kind of competition between face processing and text processing. There's no evidence yet for age of learning effects on face processing, but people who are briefly deprived early in life because of congenital cataracts never develop expert holistic face processing, even if they have years of normal visual experience. So, all right, so I summarized this already. We've started recording from some of these face and shape areas to ask, is this model plausible? So here's a monkey with an electrode track going through first a face area and then a shape area. So each row is a cell, each column is an object, is an image, and we encountered first a bunch of cells that responded better to faces, and then a bunch of cells that responded better to shapes. This was one of our adult learners, so we don't yet see any segregation of learned and untrained symbols. We haven't yet recorded from our juveniles, but their skulls are nice and thick now, so we think we can. But what we did observe was suppression to faces in this area. And suppression to faces is not what you would expect if the face area were just specialized and this area were just a general object category. You would not expect withdrawal of face selective responsiveness, but some kind of a competition model would be consistent with suppression to faces in this area. We have just recently acquired 8 juvenile monkeys who are just as cute as they can be, and we are teaching them 2 sets of symbols at different times of development. We can fit in training to 2 completely separate sets of symbols over a year each before they hit puberty, so we're hoping we'll see some differential localization in the 2 symbol sets. And lucky us, we had a baby born in our colony, and the baby sits around now and watches the mom do her video tasks, her touchscreen tasks, so that's about as early as you can get. And we don't have any idea yet how this baby is going to work out. Thank you.
So this is really just a speculative question, but
The entire last half of my talk was speculative.
Yes. So I was curious, like, how far does this extend in terms of, areas that can be trained to look as if they may have an instinctual, brain, functional specialization. In other words, what types of tasks and what types of, activities that are engaged in can be pushed so that they look like reading. Where you have this, component that looks like it's instinctual, but probably isn't. And but conversely, like, how many areas?
From low-level vision, you can put the optic nerve into auditory cortex and see with it.
It doesn't have to be invariant to different letter symbols.
It is. Did I answer your question? I don't even need to get. I tend to get low whenever I answer a question, and as far as I'm concerned, the whole brain must be plastic because every early sensory area is plastic. I bet the motor system is, but I don't know how people deal with that.
So following up that, so what areas? So as I relate to things like such as a language instinct or some kind of.
I think it is. See, I think that the universals that Chomsky and Tinker talk about are logical and biological constraints, not evidence for a language instinct. But that's my own personal opinion. Now, sorry, I want to make sure I answer this question because I really didn't answer what I thought I was going to say.
How do you think they would behave if you gave them the same symbols but in different fonts?
I know this, and they do fine. We had a serif font and a sans serif font, and it took them a couple days to get used to it, but then they were fine. And different sizes, scatter, all that color. Yes.
It's a great talk. Really, really cool. When you showed a very key piece, which is there has to be a gate function. So what's your proposed, at your lobe, at the neurons, like, how does the gate work? What's the gate?
Okay, so we have done that experiment in that you get holistic processing. So if you have a cell that responds to eye size, it will not respond to eyes unless it's in the context of an intact face. So even at the level of single cells in the middle face patch, the neuronal responses are holistic. And that's easy. Us neuroscientists just say threshold nonlinearity, and then you're done. That's all you have to say.
So but cellularly, how does that happen? So what is
Threshold
So
nonlinearity.
within a single cell?
Yeah.
Is that also a GABAergic interneuron regulating
No,
it?
it really is threshold. There's a lot of things that can happen until you hit the firing threshold, and you can add up a whole bunch of things, and then nothing happens, and then suddenly it does, and you can require certain things to be true, logical, and not gates.
So additional inputs to the cell changing its threshold?
Yeah.
So when you're thinking about connectivity of a big, so stick with your face thing, right?
Mm-mm. Let's go to
Stick with your-
orientation.
Stick with orientation. Stick with
Yeah.
orientation. So
This has all been worked out in orientation, for example. So you have narrow tuning, you don't, if you lower the contrast, you don't broaden the tuning, that sort of thing. It's all been worked out at a really threshold level, at a ion channel kind of level in V1.
So for characteristics that emerge in a bigger way, and you're compiling the bits, right? How do you put the pieces together using those models? Like everything would have to build modularly, right? So
But you can make it so the cell won't fire unless certain criteria are met.
which requires the contributions of lots of other inputs.
And you can do it at several levels too. You've got a hierarchy. Does that make sense?
I'm working on it.
Yeah. So
I mean, so for certain, it seems like the brain has, we can science out some kinds of functional organizations that are simpler, starting at the very simplest ones, and then you get the face, the more complex ones.
Yeah.
There's all sorts of ones that we don't know how to even look at or haven't found out. Brain imaging is giving us some tools to find lots of more of these little functional things, but it's finding at this much more macro level. So,
yes. So you have no handle on the thresholds that you need to get to each level.
But trying to,
Yeah.
bridge between those sort of two more extremes. What are the component pieces? What are good models to look at that you could sort of?
I would look at orientation tuning in V1. So cells that will respond only to a particular orientation. It won't respond to any spot anywhere along that orientation, but as soon as you get 5 dots lined up, it will. And some very good people using fine intracellular recording have figured out how you can do this. And usually it involves a threshold nonlinearity. And, oh God, I forgot about this, normalization. That's the other big one. Oh, so if you're just equally stimulating all the other cells around you, they'll inhibit you. So normalization and thresholds give you really fancy computations that we understand only at the lowest level. But to my mind, responding just to a face is really not that different from responding only to a particular orientation. Does that make sense? And these people are still working this out in V1, oddly enough. Only really just now, if you want, I recommend David Furster, who has figured this out, how you can apply essentially gates to a slightly complex visual stimulus and get all kinds of emergent behavior. Yes?
I have a question regarding the electrodes that you use for. So you use a single electrode for some recordings, or it's an array of electrodes?
We use single electrodes.
And so, how do you find a specific cell that fires to a specific face? I mean, do you move your electrode from cell to cell, or it's? I mean, you can probe a single.
So this is one day's recording that I did in my monkey, and this is electrode depth, and I go 100 microns every step, so I get a new cell. we correlate it with the MRI, with the functional MRI, with the anatomical MRI, and with the gray-white matter transitions. So yes, we're going to gear up and use multi-electrode arrays, but honestly, recording just many. So I have a wonderful monkey. This is like 10 minutes per cell. He works a long time.
So can it be that, for example, on that column or on that trajectory that you have your electrodes move?
These are 75 images, 25 symbols, 25 shapes, and 25 faces. And I show them really fast, 200 milliseconds each, because the cells are fast enough that, and I just sit there all day with my monkey, and he sits there and watches the screen, and the cells respond. And then I know that this was a face area because I can hear it. But you're right, we should modernize a little bit and use multi-electrode arrays, but they make big holes in the cortex. I mean, this is much safer.
If you do the same experiments for virtual reality, for more complex vision recognition.
We could, but I'm interested in shape recognition. This is a monkey who knows his letters, so I just record. You can't do a lot. You have a trade-off between a number. And we also have a recording grid up here, which is like 10 by 10 holes, so I know exactly where I'm going down each day. And I usually stay in one area for quite a while till I'm sure I know what's there. This has all been worked out for years. It's pretty standard. Not here, but it is pretty standard. Yes?
You said that in the illiterate women, the face recognition took over where the letter recognition would be in other people.
I didn't say that. I said took over. Well, maybe that's what was there to begin with. Maybe that's what's there. You know, you see faces earlier in development than you see text.
Right.
So I would be more thinking that text takes over a face area than that a face area takes over a text area. Is that okay?
Yes, but my question was, are these women better at face recognition?
I betcha.
And is there twice the amount of cortex devoted to faces?
I have a low-level answer for that. People who have amblyopia, in which one eye has taken over the entire cortex, have hyperacuity in the good eye. See, I warned you I'd do this. I always go back to B1, because that's where the rules are worked out. Yeah.
Well, what do you think about how the adults are doing this task? So they're getting perfect accuracy even though they're slower. Are they doing a completely different computation than the juveniles?
I bet. So many people say that later learned languages are more anterior and more diffuse. So less clustered. You can still do it. You can get long-range specific connections. You can do it more consciously. I mean, I can learn a Chinese character, but I'm always going to have to think about it and associate it with other things and say, yeah, it looks like a gate and it's got a thing on the bottom.
So you think it's just less clustered and you have the same
I
kind of
do. I think clustered gives you automaticity, ease of processing. I mean, we adults can learn. It's just harder. And I think we really use a different part of our brain. Yes.
Try to bring the letters.
Not yet. We're going to. Yes.
What do you think the role of basal ganglia is in this?
I have no idea. I've listened to Anne Graybill, but I don't understand the basal ganglia.
There's a literature about automaticity that comes in things that take more cortical effort, that once they're automated, less cortical effort, and there's something happening there.
That's outside my, I'm comfortably outside my area of expertise already. This is way outside. Yes?
So what do you think about the experience and being able to discriminate faces and functions in a certain way? So for example, if you're around one certain opponency, you can distinguish differences versus if you're Koreans, Japanese, South Asians versus Japanese, not supposed to do with opponencies, and wouldn't be able to distinguish.
You can learn as an adult. It's just harder.
Do you think being able to discriminate the ethnicities is
I would expect so, but they won't let me record from your face area. Yes.
Two questions. The first question is, have you looked at in the monkeys who, specifically the adult monkeys who learned the symbols, differences between the symbols and the dots? So
In that,
do they first of all respond at the same rate as the symbols and the dots? So if you compare the symbol and the same number of dots, would they show the same behavioral pattern? And then would they be using different parts of their brain?
I don't know. We actually have given most of them choices between symbols and dots, and they value them perfectly.
Okay.
Actually, the kid monkeys undervalue the dots slightly, but I think that's a Weber thing.
Mm-hmm. And
We haven't done it with the adults.
And a second question would be much more esoteric, I guess. When you look at when you talk about the faces and you notice that you have different cells responding to, in relation to what you described as almost like an ideal or an average face?
Average face, yes.
Yeah. Do you think that that might explain different perceptions of beauty in some ways? That
Sure.
What you're exposed to as a potential infant when you're first learning to discern faces might become an idealized face, and then that ends up being your search for beauty.
So what David Parrott claims is that if you take an average face and you compare the average female face to the average face, you then make a caricature of what a female face looks like by exaggerating the differences. That's beautiful. So it's actually a caricature of sexual dimorphism. And if you look at modern movie stars, you can see that they are a caricature of the difference between men, average difference between men and women. David Parrott has done a lot of research on this. Yes.
Just one comment on your observation about the properties being perpendicular to the boundaries.
Yeah.
So one implication of that is that there's a local coordinate system for the two parameters being mapped.
Mm-hmm.
That is, you can only meaningfully take a derivative, and so that there's, it implies that there's some reason why the derivative is an interesting applied.
Yes. People have modeled the V1 stripes that way. And it also explains, and I forget why, you get striped tails in spotted animals, but not the reverse. Same kinds of mechanisms. That's all. That's more than all I know about that. Yeah.
So the idea is that you have basic learning systems that are capable of producing expert behaviors or expert perceptions.
Learning. So those of us who work in low-level cortex like to think of these things of post-tetanic potentiation and functional segregation as related to learning. But learning is not, this is lower than learning.
Sure, right.
Okay.
So like the Hebbian mechanism.
Yes, okay. You wouldn't call that learning.
Okay.
Adaptation, I don't know. Okay.
I guess my question is just, is there like a class of objects that fall outside of being able to produce expertise for them?
Wouldn't you have thought that reading in monkeys
Well, I mean,
would come into that category?
I don't know if they're reading, right? Like
I don't know what
have,
they're
like,
doing
symbol
either, but
relations to.
they're really good at this. And they're responding to what you would normally consider something that's outside their normal experience.
Well, what about say like impossible objects?
Ah!
So.
Oh, heavens. This is a different talk, okay? But you can, artists have discovered that your processing of depth is quite local. It starts in early cortical areas that are local, and so what impossible objects do, and a lot of paintings do, is to have local depth cues that are globally inconsistent. And monkeys are just like people. They have local depth cues, and they will probably respond just like we do to them. So that is a manifestation of local computation, not anything important. So all these people who do ray tracing programs,